Caitlyn Cardetti from Molecular and Cellular Pharmacology will talk about what does this protein do, a case study on Neugrin.
Did you know that there are a bunch of proteins in our body that we don't even know about? Actually, biologists can't even agree on how many proteins we have. Some say 40,000, some say 400,000. Who knows? A big reason we don't know is because not all of our cells produce the same proteins, and not all of these proteins are produced at the same time. So how do we start figuring out what a protein does? Well, one way is to remove it. And that's what I did in the case of my protein, Neugrin. I got rid of it. I used CRISPR-Cas9 gene editing to create a cell line that lacked Neugrin. And then I started to look at what went wrong. I chose Neugrin specifically because I knew that this protein is required for the energy production in the mitochondria. And this energy production in the mitochondria produces over 90% of the energy for us and for our cells. My interest in studying mitochondria is because when this energy production is disrupted, it can lead to diseases like neurodegenerative diseases such as Alzheimer's or Parkinson's, or it's involved in cancers and diabetes. But back to Neugrin. I knew that Neugrin was a mitochondrial protein that affected this energy production process, but that's a very complicated process with many steps and many players. So I wanted to take a fine-tooth comb and look into what exactly it was doing and in what step. I looked at how the loss of Neugrin affects the shape, size, and distribution of mitochondria in the cell. And I also looked at the mitoribosome, which is a protein RNA complex that translates mitochondrial RNA into proteins that build up the machinery needed for energy production. And what I found was that this mitoribosome assembly was not assembling right. So this mitoribosome is made up of RNA that comes from the mitochondrial genome and proteins that come in from the nuclear genome. So I looked into both of these further, and I found out that the mitochondrial RNA itself was disrupted. So we want to know what proteins do so that when they don't do their job right, we know how to target and fix them. So in the case of Neugrin, we know that when Neugrin is not functioning right, it disrupts energy production. And it does so at a very, very early step by affecting the mitochondrial RNA levels, which then affects the mitoribosome assembly, which then affects all the other downstream processes. And we know that when this energy production system is dysfunctional, it could be implicated in many diseases. So now we can study if Neugrin itself is implicated in any of these diseases.